Welcome to my presentation on the regulation of glycolysis and glucaneogenesis process and how aldolase expressed in both processes. Our agenda today is going through the overview of glycolysis and glucaneogenesis process and how aldolase is involved in both processes and the different isoforms of aldolase and their different expression pattern. And the last point is examination of how aldolase B expression can be regulated by transcription factors. So let's have a look and learn what glycolysis is. Glycolysis is the process of breaking down glucose into pyruvate molecule. This is happening in the cytoplasm of the cell outside the nucleus, and it generates ATP molecule to support the cell with energy. Let's have a look for this process. So in this process, we have glucose molecule, and this glucose molecule present in all our body cells and tissue, and this glucose, which is C6H12O6, is cleavage or breaking down into two molecules of pyruvate. During this process, when we have the molecule, glucose molecule, and we breaking down this 6 carbon into two molecule of 3 carbons, which is pyruvate, we generate 4 molecule of ATP. Of course, this process, as we explained before, is take place in the cytoplasm. On the other side, glucaneogenesis, this is the process that we convert the or form glucose from different carbon substance like fat and protein, and we make the glucose by the end of this process. So during fasting or starvation, our body need energy. So we can get this energy from amino acid or protein and fats and support our body with energy in the form of glucose molecule. This fat or protein is not necessarily just only present in the liver, so we can find it in the kidney or different body parts like brain, like small intestines, large intestine, and the brain. That was overview for glucose and glucaneogenesis. The second point is how aldolase is involved in both processes. First of all, we need to know what is aldolase. Aldolase is the enzyme or protein that involve in both process and play a central role in interconvention of sugar molecule. But how this happen? In process of glycolysis, aldolase start to cleavage the glucose molecule and the fructose 1,6-phosphate or biphosphate into two molecules of 3 carbon. The first molecule is phosphoglyceraldehyde and the second molecule is dihydroxyl acetone phosphate. So dihydroxyl acetone phosphate and phosphoglyceraldehyde 3, we cannot find those molecules without presence of aldolase. So aldolase is required to form this molecule. But what is the importance of aldolase in this process? Aldolase is play an important role in a catalyze and cleavage the six carbon into two molecule of three carbon to support the body with reaction and support the body with energy and can be further processed to produce ATP and NADH. But what is the role of aldolase in the second process, which is the reverse of glycolysis? In glucaneogenesis, this is happening in the liver, so we can make the reverse over here. So we combine the molecule of fructose 6-phosphate together with phosphoglyceraldehyde and uh, dihydroxyl acetone phosphate. We combine them together to form the glucose molecule and store this molecule during any kind of starvation or any body need or require for energy. This is mainly happen in liver. So without aldolase, we cannot combine the three carbon molecule to form glucose, which is 6 carbon. The third point we need to talk about it is different isoform of aldolase and their different expression pattern. All of us, we know now that aldolase has a vital role and it is a vital enzyme and protein in the pathway of glycolytic pathway. And like other protein, it's found in a different isoforms and variety patterns of tissue-specific expression. But the word isoforms mean different version of protein that are produced from the same gene, but they have distinct structure. So, for example, we can find different form of aldolase, but aldolase A, B, and C, all of them have the same structure, but have different function. For example, aldolase A, which is the first isoform, we can find it in a muscle, and it has the same structure of all aldolase, but the presence of it in a muscles. So the role of aldolase over here is represented by muscle tissue or localized in the muscle tissue and erythrocyte. Erythrocyte is the red blood cells and it participates in the glycolysis in all our body tissue. The second isoform is isoform aldolase B, and this aldolase B is present in the liver to regulate the role of detoxification fructose. And we're going to talk about aldolase B in details in the next slide. The third form of aldolase is aldolase C, aldolase isoform C, and this is present in the brain and has an important function in the brain energy and production needs and any neural transmitted and in a different kind of neural development, like interact with the cytoskeleton component and play an important role in the neural function. So in a brief, the difference between aldolase A, B, and C, the location of them. So the location of aldolase A in the muscle, erythrocyte, and embryonic tissue, B in the liver, kidney, and the small intestine, and C in a brain and central nervous system. But the primary role of each aldolase A is a very important glycolysis, aldolase B for glucaneogenesis, and aldolase C for energy metabolism in neurons. The side effect of lack of these isoform can cause different kind of disease. For example, if we lack aldolase A, so we can find hemolytic anemia. If we miss aldolase B, so we can find different problem in heredity function. And for aldolase C, if we missed it in our body, so we have neurodegenerative disorder. Here we can talk in details about the aldolase B and how aldolase B can be regulated by transcription factor. As we said in the previous slide, we talk about aldolase B as a kind of enzyme which play an important role in heredity. So aldolase B play an important role in glycogenesis and fructose metabolism in liver. But how these transcription factor regulated the aldolase B? Let's see how many kind of these transcription factor. We have four important transcription factor. The first one is hepatocyte nuclear factor 1 and hepatocyte nuclear factor 3, CEBB and DBB. Hepatocyte nuclear factor 1 is required for liver function, has a very important role in the liver function, as aldolase B is the one of the gene influenced by this transcription factor because it directs the expression of the number of liver-specific genes. For hepatocyte nuclear factor 3, this is very important to activate or stimulate aldolase B in reduction and in the mini DNA to reduce the rate at which aldolase is produced. For CEBB transcription factor and D-side binding protein, so aldolase B is very important in regulating the metabolism, differentiation, and immune system. So without this transcription factor, so we can find different problem in our immune response or in our metabolic function because aldolase B, by using this kind of transcription factor, is expressed the level of appropriate for metabolic demands such as during glycolysis process and glucaneogenesis. For D-side binding protein, it's very important in our biological clock and circadian rhythm, which are regulated by transcription factor that link gene expression to the body daily cycle by binding this aldolase B during a specific time of the day. The problem now, what happens if this transcription factor is not working with the aldolase B expression? If there is no interaction between aldolase B and these transcription factor, of course, will affect our heredity and make mutation to our body, in addition to different kind of autoimmune system and metabolic process will affect negatively. And of course, this is also effect on the gene expression. So we cover all the 4 points at the first beginning. In our agenda, we talk about what glycolysis, what gluconeogenesis, what aldolase, and how it's involving the two process, how it's formed the glucose in this process, and how it's breaking down the glucose in the glycolysis process, and different form of isoform aldolase A, B, and C, and the function of each. And last but not least, how the expression and the transcription factor affect the aldolase B in our body tissue. Thank you so much for listening and watching my video, and I hope you enjoy the lesson. These are the reference of my presentation, and thank you so much for listening.